Hello guys, welcome to Boson's Botany. In today's lecture, I am starting with receptor tyrosine kinase, commonly known as RTK, under the topic cell signaling. In my last video, I have dealt with GPCR, which is G protein coupled receptor, and I have told you that there will be a series on cell signaling. So this is my second lecture on cell signaling. So let's begin. So what is RTK or receptor tyrosine kinase? So before I dive into the structural definition, let me give you an overview of this signaling mechanism. RTK is needed in different cellular processes, including growth of cell, motility of cell, differentiation, and metabolism. So basically, RTK is helping a cell to survive and it keeps the cell alive. And if there is any abnormality in this signaling process or the receptor itself, or the downstream signaling molecules, which will be dealt in the upcoming in this lecture only, but subsequently I will deal with all those signaling molecules. So if there is any abnormality in either the receptor or the signaling molecules, then there will be cancer. And why cancer is initiated if there is any abnormality, that I will define while explaining the signaling process. So let's start with the structural definition. So the structure of RTK is very important because questions are often asked from this part as well, and it is quite simple also. So as you can see, there is a ligand. Let me write here the ligand. Let it be a growth hormone or a growth factor. Growth factor. A ligand could be anything. It could be a hormone. It can be a growth factor. It can be anything. But in RTK, there are growth factors which are actually participating maximum. We get epidermal growth factor. Then we have the platelet growth factor. There are various growth factors which are initiating this signaling pathway. So let me take this example, growth factor as our ligand. And before this growth factor binds to the receptor, as you can see, they are far apart. Okay, there are two units of this receptor. They are far apart before the ligand binds. And this unit, as you can see, this is similar. There is no difference in between these two units of RTK receptor. So this receptor will have one external N terminus, which is towards the exoplasmic site, and there is a transmembrane domain. This is the transmembrane domain which is having alpha helix, and alpha helix is basically required to make the transmembrane domain so that this portion can be embedded inside the plasma membrane, right? Because transmembrane domain has all those hydrophobic amino acids which are required to adjust with the hydrophobic environment of plasma membrane. As we all know, plasma membrane is composed of lipids. So lipids are non-polar in nature, they are hydrophobic, so they need hydrophobic amino acid to allow this transmembrane domain to be embedded. So this is the transmembrane domain having alpha helix. And towards the cytosol, there is the C terminus, which is the actually most important part of this whole receptor, because this C terminus will have tyrosine kinase domain, and this tyrosine kinase domain will initiate all the downstream signaling processes after the ligand or the growth factor binds to the receptor. So after the receptor is bound to the growth factor, which is the ligand here, there is dimerization. As you can see, these two units of the receptor will come closer and they will form a dimer. This is dimerization. and this will allow further process to happen. So after the receptors are dimerized, what will happen? This C terminus will autophosphorylate each other. As you can see, this C terminus is autophosphorylating this one, and this one, that is this left hand side C terminus, will autophosphorylate the right one. So there is autophosphorylation, and this autophosphorylation is the most important part to make this receptor active. So this receptor is now active. Receptor is active and it is ready to perform all the downstream processes. So this is the overview of the structure of RTK, and questions are often asked from the structural point of view. So just keep these points in mind, and there are the points written here which I have already discussed. As you can see, receptor autophosphorylation, then cross phosphorylation is also there. Okay, so there will be both autophosphorylation as well as cross phosphorylation. Cross phosphorylation means uh, this part, that is the right hand side C terminus, will cross phosphorylate this one, and this one will cross phosphorylate this C terminus, and there will be autophosphorylation as well, that is phosphorylating itself. And there is transmembrane alpha helix. Everything is written here, so you can go through these points. So I hope the structural definition is clear. Now there are two exceptions. First, insulin receptor is already dimerized even before the signaling molecule is bound to the receptor. So this is very important. Receptor which is needed for conducting signaling when insulin is the receptor signal. Insulin is the signal and the receptor for insulin is insulin receptor. So this receptor is already dimerized. It is already dimerized even before the signal is bound to the receptor. And the second exception is EGF, that is epidermal growth factor receptor, does not show autophosphorylation. Okay, so this is again very important. These two exceptions are quite often asked in the questions. So just keep them in mind. Now, there is a very important domain which is known as the SH2 domain. So what is this SH2 domain? SH2 domain binds to specific phosphotyrosine containing peptides of the activated receptor. So what does that mean? After the receptor is activated, that is the ligand is now present on the receptor is dimerized, there is both autophosphorylation and cross phosphorylation on the C terminus on the tyrosine domain. Now what will happen? Now we need to have the downstream signaling, or the process should be initiated. For that, this downstream signaling molecule will bind to the phosphorylated domain of the receptor which is having SH2 domain. This domain should be present on the downstream signaling molecule, and this domain will actually bind through this phosphorylated part. As you can see, it is very clearly shown here. This is the SH2 domain that will bind to this phosphorylated part of C terminus, and using this SH2 domain, this downstream signaling molecule will bind, and there will be signaling initiation. I hope this is clear, and on both the sides, as you can see, there is downstream signaling molecule 1 and downstream signaling molecule 2. On both the sides we will have the SH2 domains. So SH2 domain is basically required to bind the downstream signaling molecule by attaching on the phosphorylated site on the C terminus. I hope this is clear. So there is again one exception. Insulin receptor has IRS1 site in place of SH2 domain. So insulin receptor will have IRS1 site, not SH2 domain. So this is the third exception you need to keep in mind. So I hope this is clear. These are very small points but extremely important. So just note them down. And now let's begin with the signaling. It is again very important. So this growth factor, which is the signal, this will bind to the receptor, and after the ligand receptor binding has been completed, there will be cross phosphorylation and autophosphorylation on the C terminus, as it is very clearly visible. There are phosphorus attached. Now what will happen? All the downstream signaling molecules will be activated. So first will be RAS. RAS protein will be activated by attaching a GTP molecule, and this is phosphorylated. Now this RAS protein will further phosphorylate RAF1. RAF1 will phosphorylate MEK. MEK will phosphorylate ERK. So these all are basically kinases. These are kinase proteins, and this order is fixed. Okay, it means RAS is the first one and ERK will be the last one. You cannot change the order. Now what are the signaling that will be initiated in RTK signaling process? So as I have already told you, growth and cell survival, and there will be no apoptosis. Apoptosis means cellular death or killing cell. Okay, apoptosis means killing the cell. So apoptosis will be inhibited, that is apoptosis will be stopped by this signaling process and growth will be initiated. So as I have already told you, if there is any abnormality in RTK signaling, then cancer will happen. So I think this is making some sense now, because cancer is basically uncontrolled cell division, and cell division leads to cellular growth, cellular survival. So if something is like highly active in this signaling process, for example RAS is overexpressed, that is the functionality of RAS is highly expressive, RAF is extremely like highly expressed, MEK, ERK, they are functioning much more as they are required. So if there is overexpression seen in case of all these proteins, then cell survival will be extremely like demandful, and cell growth will be initiated, and for that reason apoptosis will happen but very less, and this overexpression will cause cancer, that is growth will manifest or it will increase, leading to cancer, because as we all know cancer is uncontrolled cell division and cell division leads to growth. So if any of these proteins are overexpressed or they are hypermutated, then cancer will happen. So if a cancer patient is having any of these proteins hypermutated, then we will prescribe them with anti-cancerous drugs. For example, there are already mentioned these are some anti-cancerous drugs that will inhibit the receptor itself, RTK inhibitors. Now this this is another pair of anti-cancerous drugs inhibiting the RAF protein. Again, these are anti-cancerous drugs inhibiting RAF. So we will give anti-cancerous drugs so that cancer is not happening to that patient or that patient is saved, and basically we are stopping cell growth or cell survival and initiating apoptosis. So I hope this is clear. This is very important because cancer is basically due to multi mutation. There is, if there is only one mutation, then cancer will not happen. It has to be multi dimensional mutation. Means if RAS is mutated, RAF1 has to be mutated, MEK has to be mutated. So then only this process will be like increased and inhibiting apoptosis and leading cancer to progress. So RAF or RAS belongs to GTPase superfamily of intracellular switch protein, and it is known as oncogene. So RAS is basically a GTPase and it is an oncogene. Oncogene are genes which promote cancer. They are cancer promoting genes. So you might be thinking, then what is the necessity of having RTK signaling? It is not good for our health, right? But everything has to be done under control. Growth is also important, apoptosis is also important. So if either of these processes, that is growth and apoptosis, if either of these processes are uh, happening too much or too less, then problems will arise. If growth is happening like without any control, then there will be cancer. If apoptosis is happening way too much, then there will be other problems. So everything has to be checked and everything is important. But if one process exceeds its own potential, then such ailments or health issues can take place. So that is why RTK is very important. So I hope this lecture gives you some insight into RTK, and let's start this series by reading all your comments. Just write down what else you want to study in signaling, cell signaling, and I will try to make videos on those topics. So I hope this video helps you. Thank you.